welcome Dr. David Evans here, who is interviewing for our developmental science senior level position. And Dr. Evans is currently a professor at Bucknell University. He received his PhD in developmental psychology from Boston University and completed postdoctoral research fellowship in neurodevelopmental and neuropsychiatric disorders at the Yale Study Center. Dr. Evans is currently senior investigator and director of the Laboratory of Developmental Neuropsychology at the Geisinger Bucknell Autism and Developmental Medicine Center. Dr. Evans' research focuses on understanding the interface between normal and abnormal development. His work links genes, brain, and behavior as integrated systems in the study of both typically developing individuals as well as those with or at risk for neurodevelopmental or neuropsychiatric disorders. He has specifically repetitive behavior and restrictive interests in children using genetic, electrophysiological, and neuroimaging techniques. And Dr. Evans currently has funding from the Simmons Foundation for autism research, as well as funding to help build the Autism and Developmental Medicine Center. He has an impressive number of publications published in journals such as PLOS One, Biological Psychiatry, Behavioral Brain Research, Brain and Cognition, Child Psychiatry, and Human Development, Child Development, and Lancet Neurology. His research interests are interdisciplinary and they lend themselves to several levels of analysis that could foster collaborations across disciplines here at UMass, specifically within the department. Here, his work connects to researchers in the Developmental Science Division, the Clinical Division, the Cognitive Area, Behavioral Neuroscience Division, and the Neuroscience and Behavior Program. And he would contribute to our neuroimaging group, our emerging early childhood focus group, and the Center for Research on Families. And today, he is going to talk to us about copy number variation syndromes and neurodevelopmental disorders. So please join me in welcoming Dr. Evans.
Thank you. Thanks very much. Thank you. It's great to be here. I appreciate the invitation. I'm happy to have met the people I've met so far. Looking forward to meeting the rest of you, or at least some of you. I've got a lot to present today. Challenging talk to prepare for because I'm really thinking about it in two major pieces. The first part, really things that I started as a postdoc, still continue to do, comprise most of the body of my work for the last years or so. And then I'm going to shift and I'm going to talk about work that I've really focused on looking at the copy number variant syndromes and looking at behavioral phenotypes in certain CNVs. And that's work that I've started to do collaboratively in the last 3 or 4 years and really where I see myself going. And I'm hoping to be able to kind of weave a thread of continuity between these two parts of the talk. Some of the first part, I'm going to fly through screens because I'm worried about time and because I'm afraid you'll ask me questions I can't answer. Not really. But I'm going to talk first about some of the work on repetitive behavior, just very cursory, brief studies that have, some studies that have happened recently, some that are ongoing. And then I'm going to present a model. Lisa mentioned this paper in Lancet Neurology, and the colleagues and I presented a model that is really going to be kind of a guiding light for me for my work, I think, moving forward, that looks at the behavioral phenotypes in CNVs. One of the things, grad student even, that was kind of the mantra of my early research and interest was this idea that there's an interface between normal development and pathological development, that the historic division between clinical syndromes and normal development is an artificial one. And in fact, that we can really learn a lot about normality by studying typical development and vice versa. And this is what really resonates in my work today as well. We're all pretty familiar, I think, with the traditional diagnostic approach in psychiatry, but this has also been, this is a legacy, as I talked to some of you about, of a couple of centuries of taxonomy in science, of categorizing and classifying that has served some important functions, but in some sense it's kind of gotten out of hand. So when we think about a psychiatric diagnosis or a neurodevelopmental disorder like autism, for example, we generally think that you've either got it or you don't, that the diagnostic thresholds are reached, the person has reached the criteria, diagnosis is conferred, treatment plan is given. But the reality is that there's more of a distribution, that almost any disorder that we think of can be conceived as having quantitative dimensional traits. I put this in here fully recognizing that most of us do not function out here, but I didn't know how to crop that properly. But you get the idea, is that most of us are in the space. Some people have few symptoms, some people have many symptoms, but largely what we've done is take quantitative and erect these arbitrary distinctions. So we've got a perfectly nice quantitative trait like IQ, which is on purpose normally distributed in the general population, and yet using standard deviations, we've erected these perpendiculars that determine and that demarcate and separate people who are above a given threshold from those who are below. And I recognize that the current intellectual diagnosis for intellectual disabilities between 70, 75, so they've kind of gotten funky and quantitative there. But really, there's been a long history of you've got it or you don't, even where it concerns quantitative traits, which is really, in a way, a misleading and a misuse of what could be really powerful data. What I'm going to talk about is for the next few minutes, is work looking at, in some ways, autism spectrum disorders, but it transcends that. I'm really interested in dimensions of behavior, and I'm focusing particularly here on this class of repetitive behavior and restricted interest. So these are the two diagnostic criteria for ASD, according to the DSM. Historically, the amount of research effort understanding the social deficits in ASD has really far outstripped the work that's been done on the repetitive behaviors and restricted interests for whatever reason. The other thing, what I want to talk about, some of the issues here, is that when we talk about autism spectrum or anything like autism spectrum, it's really important to recognize that there's not just a continuity between the normal manifestations of behavior and clinical manifestations, but there's a tremendous overlap across different diagnostic categories. And in fact, the rates of comorbidity in neurodevelopmental and neuropsychiatric disorders are astronomical. And this is not an exaggerated estimate, that comorbidities in ASD generally exceed about 90%. It begs a lot of questions. At least one of them is, are these really different diagnoses, or are we looking at a series of overlapping continua of behaviors that happen to co-occur in individuals? And that's some of the stuff that's driving my current work now. We see it also in a series of different neurodevelopmental disorders that have been characterized genetically. Okay, so in fact, for these are just a sampling of Prader-Willi syndrome, Cri-du-Chat, Smith-Magenis, Williams syndrome. These are disorders that have been linked to intellectual disability and to autism spectrum disorders. They confer increased risk for both. But what's interesting about them is that all of these phenotypically are marked by really interesting patterns of repetitive behavior. In other words, what marks them as the syndrome initially, phenotypically, is kind of interesting and odd patterns of behavior. So Prader-Willi syndrome people do a lot of excessive skin picking. In fact, most of them meet diagnostic criteria for OCD because they ruminate about food and they're generally morbidly obese and they can't stop eating. They're like compulsive eaters. Cri-du-Chat has this characteristic crying sound, but that's what marks Cri-du-Chat. Smith-Magenis is a disorder, genomic disorder, where the people engage in a lot of self-hugging, particularly when they get excited. And they just kind of grab themselves like this. And sometimes they hug other people, but it's not a social hug. It's like kind of squeeze the breath out of your lungs kind of thing. And so the repetitive behaviors that I'm going to talk about also kind of map out on some that I'm going to present later on. This is an intentionally busy slide. And all I'm saying is that when we think about what repetitive behaviors or what can be included or subsumed within the class of repetitive behaviors, it's really wide. We could talk about stereotypic motor movements, self-injury, all sorts of self-directed behavior, a whole group of tics, compulsions, focused interests. And these things are really highly overlapping, wide-ranging, and interestingly, many of them are very, very common in typical development. And this is what I'm going to focus on a little bit. In fact, even some of the severe ones, I mean, so these are some of the more benign, but anyone who's got or interacted with a 2-year-old or a 3-year-old recognizes some of these things. Rigid adherence to rules about bedtime and mealtime, stories of, a child refusing to eat because the parent cut the sandwich crosswise instead of the other way, or peas roll into the mashed potatoes and it sets off a real firestorm. They need things to be the same. They've got a kind of a cognitive sense or a schema of how the world should work, and when it violates that, it creates problems. It creates a lot of subjective distress. And these are all typical. What's that?
I said few.
Okay, yeah. Anyway, so these are some of them, but there's preoccupations with symmetry, attention to minute details, etc., sensory sensitivities. A lot of the things that you see in neurodevelopmental disorders like autism are very much present in typical development as well. Here's a thing. I don't want it in the Little Mermaid cup, so obviously he's made a request in the middle of the night, and rather than sort of appreciating the overall goal, the focus is on some kind of circumscribed interest and attending to something that's not how I pictured this, The overall goal of getting the water's great, but you did it wrong kind of thing. This is the sort of behavior that's really common in typically developing preschoolers and faculty. Without going into too much history, but a lot of the work was driven theoretically when I looked into this work as a postdoc. So as a developmental psychologist, am a developmental psychologist, did my postdoc at a place that has clinics for OCD and autism and Tourette's. And when these families started to come in and say, oh, the kid's really, preoccupied with, I remember one kid had a G.I. Joe doll and the paint on the eyebrow was off and the kid sort of flipped out. And the parents are in there as a clinic saying, does my child have OCD? And I remember thinking, boy, a lot of these behaviors for a 3-year-old and a 4-year-old, our early identification is great, but these are really, really common in typical kids, and what's going on here? And looking it up, there's one paragraph that Arnold Gazelle wrote about this and said, we don't know anything about it. But these kids go through a ritualistic phase at about 2.5 years to 3.5. It goes away. We don't know why the hell it was ever there. And it's strange and interesting, but we don't know much about it. Piaget, in his own way, talked about repetitive behaviors as kind of, reflexes and primary and secondary circular reactions and things like that. But also, interestingly, like the perseverative behavior, such as the A not B error, where motor memory basically in habit sort of trumps or supersedes knowledge about the placement of an object. And Heinz Werner, who was kind of my favorite old developmentalist, actually did a lot of talk about ritualistic behavior and magical thinking, and the link between some of these behaviors and not autism, but actually schizophrenia, which is a story I'll get into later. And Esther Thielen is someone who, much later than these grand theorists, who really was interested in motor movement and in kind of coming up with a human ethogram for what constitutes normal repetitive behavior in infants. And so I started to read this literature, and I thought, boy, all of these people are touching on it, but we still had no standards for it. We still had no sense of how much is too much, or how common is this? When is the onset? When is it supposed to fade? So when a parent says, is this normal that my child is, lining up his or her toys over and over, or has to, have the sandwich cut in a certain way, when does it cross the threshold? And the answer is, without knowing norms for this, you don't know the answer. And that was sort of problematic. So my initial work really was about trying to understand the prevalence and the development, and then later some of the correlates of repetitive behavior as a typical phenomenon, thinking that maybe we can inform these kinds of behaviors in psychopathology. So the initial study was a large-scale survey of families in New Haven, Connecticut, all typical kids. We went through the DSM to look for repetitive behavior in autism and in OCD. We kind of cast them in a normal developmental framework and then sent them out as part of a MacArthur study with Steve Resnick, who's down here, who had the MacArthur grant at that point. We did I took the data. As the data came in, I did kind of an exploratory factor analysis on half the sample, did a confirmatory factor analysis on the second half of the sample, and really came up with two factors, two principal components from the exploratory part that really mapped out on repetitive behaviors, and then this kind of sensitivity, this focus on detail that we called the just right, where things have to be just right. And these are the factors. I should say that the measure's been used and translated into a couple of other languages, and some cultures replicate the two-factor structure and some don't. So that's a confession. Next slides, I'm just going to flip through a couple of items to show you what's kind of akin to prevalence data based on ages of these items, that what percentage of parents endorse that the child does these items in this typical sample. One thing I'll say is that this sort of trend of increase to the second and third year and then a leveling off was really uniform across most of the items. We've done longitudinal and cross-sectional work. I just renormed this and expanded this, and we've got data from 1-year-olds to 18-year-olds and a companion for adults, and I'll get into why we're doing that later. But we see this continue to tail off, and it hovers around the 10 to 20% for pretty much ever. But insists things done in a certain way, engages in elaborate bedtime routines, lines up objects, you get the idea, concerned with dirt, cleanliness, or neatness, aware of imperfections in toys, clothes, etc. Okay? Real quick, in order to sort of study this, one of the things we needed was to develop this measure, right? There wasn't such a measure. But importantly, and I wasn't sure where to talk about this, and I'm sorry that these are SPSS, but my friend Heidi said, hey, at least they know that you're dealing with real data. But one of the things here that's key when we're studying subclinical phenomena of any kind is the sensitivity of the metric. And this is not to pick on it, but this is the measure that was developed by Jim Bodfish, which a lot of people consider the gold standard measure for assessing repetitive behaviors in kids with autism spectrum. This is based on 600 children from the AGREE data set. These are siblings of children who've been diagnosed with autism. And when you administer them to the non-clinical siblings, you get a floor effect. Okay? So it may be a decent measure in terms of differentiating or discriminating clinical repetitive behaviors from non-clinical, but in terms of looking at shared variance of the non-clinical versions of the repetitive behavior and, for example, neural structures or functions, this is going to fail. This won't work because you need variance in order to find shared variance. And this is our measure on a similar sample of 1 to 6-year-olds. So these are all little kids, comparable groups, but you see the distribution. Anyway, real briefly, when we kind of got into this, the next question were, well, why is this stuff there? And we sort of kept clicking the screen back to the clinical literature and said, gee, in a lot of disorders like Tourette's and autism and OCD, there is a burgeoning literature looking at executive function deficits and attentional shifting and cognitive set shifting and response inhibition. There was some work linking these disorders to atypical cortical activity, looking at things like P300 and an oddball task. The studies that generally looked at this did what we're very used to, which is take an extreme group with a disorder and a group that's been with exclusionary criteria that are kind of hypernormal and find that there are group differences, establish that repetitive behaviors are linked to cognitive domains that reflect faulty neural processing, basically. That basically the brain of someone who exhibits clinical levels of ASD or Tourette's or OCD are essentially diseased. And what we're sort of saying is, no, a lot of this depends, we're looking at mapping out brains and behavior on each other and looking at shared brains. And if you're going to look at extreme groups, you're going to find group differences. We were interested in more the shades of gray or the kind of pastel version of this stuff. Really quickly, and then looking at some of the structural and functional images, there's major findings implicating repetitive behavior and perseverative behavior to the orbitofrontal cortex, dorsolateral prefrontal cortex, anterior cingulum, and the striatum, particularly the caudate, the putamen, the globus pallidus. And so all of these have been laid out in saying, yes, extreme autism group differs volumetrically and in terms of function from a typical group. But again, we're sort of interested in looking at the shared variance in the typical sense. Well, I'll get to this later, I guess. What we found is basically a little bit what we didn't. But basically, in a nutshell, that perseverative errors on a set shifting task were related to repetitive behaviors for younger children, but not for older children. And we had this cut off at 6 for a couple of reasons. One was median split. The other was what some refer to as the 5 to 7 shift in cognition, but that's kind of old school. We also found that on a go, no-go task, just click when you see every letter of the alphabet except the letter X, looked at perseverative errors and ritualistic compulsive-like behavior in typical kids. And interestingly, the same kind of something similar. For younger children, we found an interesting trend where the errors of commission on continuous performance tasks were positively correlated with repetitive behavior and restricted interest, but the direction went the other way for kids 6 and older. This was a bit of a conundrum. I don't want to go too much into it, but the interesting thing was the fact that these are typical kids in a way. And there's a sense in which our interpretation was some compulsivity. This may also be kind of a good news thing. A little bit is good. A little bit has a kind of a guiding structure to our lives, and that may be happening there, but I don't mean to overinterpret. The other thing that we looked at was kind of testing the a P300 A lot of the ERP work in pediatric OCD and in autism was in auditory, and we were really kind of interested in visual system because a lot of the symptoms or behaviors that we're looking at are very visually kind of loaded in terms of lining up objects in straight lines and sensitivity to symmetry and things like that. And so I created this P300 paradigm. It would look something like this. That's the standard stimulus. They see this 80% of the time, and it would flash like this, and then bang. And we look at the oddball. We look at the P300 in terms of amplitude and latency to the 20% of the deviant stimulus relative to the 80% of the standard stimulus, and we actually found the latency difference, that there was a significant correlation in shared variance. That is, the people who engaged in more of these just right kinds of behaviors actually processed that asymmetrical stimulus faster. The latency was, on average, less than 300 milliseconds, okay? And it was a pretty strong correlation. I should say that that's also not just for that task. We had a control P300 task of 2 spheres, and it predicted, we regressed that out, basically. So this was the variance left over, the R-square change from the lines task or the symmetry task. In very recent work, we're doing something similar with functional and structural imaging. We're finding that when we can capture the normal range of repetitive behavior, we're finding, again, shared variance in volumetrics of the orbitofrontal cortex, and interestingly with caudate volume, not necessarily absolute volume. There's a typical right greater than left asymmetry that generally shows up in the caudate, and we're finding that deviation from right to left asymmetry in typical subjects was strongly linked to the rate of repetitive behavior and restricted interests. I don't want to talk about this, but it's also something else that we've been doing, looking at SNP data in oxytocin receptor genes. But if there's time, I'll talk about that. So I'm right about at the end of where I needed to be for phase 1, for putting up what we found. Our work, repetitive behavior, restricted interests are really common in childhood. Phenotypically, they seem to share a lot of features. It seems to be a developmentally characterizable, if I can say that, trajectory. And when you look at the studies that demonstrate neuropsychological, psychophysiological, and neural deficits or differences linked to repetitive behavior, what we're demonstrating is that we see those things linked in the normal brain and the normal behavior. And that's kind of the important take-home message, is that these are from one to the other, and we're really talking about overlap of behavioral dimensions, not characterizing pathologies as being distinct from normal manifestations. Okay. In the last few years, a couple of years ago, we were fortunate to have helped start a research clinic for kids with neurodevelopmental disorders in central Pennsylvania, and this is what we call ADMI, which is the Autism and Developmental Medicine Institute, center, whatever, but we call it ADMI. And here I've been working with a team of people that I'm sort of on the behavior, I'm doing some of the ERP stuff, getting more and more into the functional and structural imaging, and I'm working with a team of geneticists. And one of the things we're doing is looking at the impact of genomic copy number variations, which for those familiar, I'll explain that a little bit, on quantitative behavioral traits. And the quantitative part is everything that I've been presenting so far. This is also very consistent with the NIMH RDoC perspective of trying to get people to move away from the artificial dichotomies of diagnosis and focus on quantitative dimensions. So we're linking genomics and behavior. We're also, as I have done a little bit and am continuing to do, linking some of these quantitative traits to dimensions to brain structure function. And then to sort of map out and create neurophenotypes, that is kind of possible neural profiles for different copy number variations that are going to help us understand the structure and function of the brain as it feeds in or subserves certain behaviors. A copy number variation, for those who don't know, and if everyone does, I apologize if I'm talking down to you. These are structural variations in segments of DNA. So, and there can be several versions of this. So these are cartoon versions of a chromosome. There's a break in the chromosome, and when this is deleted, we have a deletion. We can determine the length of that deletion based on the number of single nucleotide pairs. Okay, and they can go from, well, technically, you can have a copy number variation that's a whole chromosome aneuploidy. That's the loss or a gain of an entire chromosome. But like Down syndrome or Turner syndrome. CNVs, though, are generally more specifically regarded to smaller regions of DNA. You can have deletions. You can also have duplications, where there's a break here, and then it's actually two copies are made. And so you have some extra material. There are inversions that occur. There are translocations where a part breaks off, shows up on another chromosome entirely. And all of these are kind of distinct versions of copy number variation symptoms. In our recent work, and I'm sorry, but I was so proud of that Lancet paper. I had to put it up there. And they gave us the cover. And David Ledbetter said, oh, it's where the child is a couple of steps behind where he should be. And we were all kind of figuring out, what is that? What does this mean? I was like, the cobbler's son goes without shoes? What? I couldn't think of anything. Anyway, one of the things that we're doing is looking at, is really trying to look at a genetics first or genomics first approach to understand behavior and the links between brain and behavior. The idea being that looking at clinical diagnosis based on the phenotype is misleading for a couple of reasons, some of which I've already mentioned. And that really the diagnostic system is inconsistent and largely incompatible with the biological reality of how most diagnoses play out. Okay? And one of the things that we say in this paper is that, and there's evidence to support, that the same clinical diagnosis may result from many, many different genetic abnormalities. For example, all of these 3 children have a diagnosis of autism. But they've all got different kinds of autism. This child has Fragile X, which is an excessive number of CGG repeats on the X chromosome. This person with autism has 16p112 deletion syndrome, which I'm going to be talking about more. This child has Prader-Willi syndrome, which is a deletion in this region on the 15th chromosome. All right? Same disorders. Maybe similar clinical presentation, deficits in communication and social, excesses in repetitive behavior, restricted interests, probably some intellectual disability for most part, maybe some other, ADHD as well, blah, 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 but certainly autism likely, okay? But for very different reasons. And this ends up being really important because when we think about treatment, You want to know what the cause is. I always say to my students, imagine you've got 4 people who present with a cough, and one of them has emphysema, and the other has lung cancer, and the other has a cold, and the other has a phonic tic where they have to, cough and clear their throat. We would never presume the same treatment for those 4 kinds of people, even though the behavioral manifestation can look quite similar. And same here. There's no reason for a drug trial study, for example, that it should work equally here. There are radically different mechanisms underlying their autism. So we want to know what the underlying is. The other thing that we find that's kind of interesting is that the same copy number variant can result in very clinical disorders. Or the same copy number variant, including all 3 on that previous page, can appear to have no disorder at all. So some people with those CNVs can appear unremarkable. They're alive and well in the general population. And here kind of lies the rub. But first, here's an example. So these are very common, well, these genetically are considered rare variants. So the most common among them is about 1 in 4,000-ish, okay? But they are considered rare variants in the strict sense, but they're the most common of the rare variants, if that makes sense. Anyway, all of these things have been associated at one point in time with intellectual disability, developmental delay, higher rates of autism spectrum disorder, higher rates of schizophrenia, higher rates of epilepsy, higher rates of ADHD, and the list could go on. So the same CNV in one person, the person could have an intellectual disability but not autism. Another person with the exact same CNV could present with autism but be above the cutoff intellectually. Okay? And this is kind of interesting and confusing and yet exciting for a couple of reasons. Here, this is part of our, this is from the Lancet paper as well. One of the things that geneticists talk about is this phenomenon of incomplete penetrance. And basically, I already said what it is, which is anytime you've got a copy number variation, any genetic mutation that doesn't confer a disorder 100% of the time, geneticists will say that that confers incomplete penetrance. Okay? And they sort of say that in a pejorative way, almost like, well, you can study that, but that's obviously not the key to autism because some people with that CNV don't have autism. So it's incomplete penetrance, so you may be barking up the wrong tree. That's sort of the parenthetical way of thinking about it. And so these are 2 CNVs. This is just hypothetical or theoretical propositions here. One, the deletion 22q11.2. and second 16p11.2. And what we see is the general population, we're talking about full-scale IQ, and we'll generally maybe see a shift from the general population on the 16p, where some of them are going to fall into the intellectually disabled range, and same here for 22q, but some of them don't. And it sort of begs the question, okay, are these CNVs incomplete penetrance for intellectual disability? So this proband here who may have an IQ of 95, does that mean that they are unaffected? They've got the 16p11 deletion syndrome,